Let's say we have this adorable raccoon, and we want to silence a gene responsible for giving the raccoon a striped tail. How would one go about this? There are two ways to silence a gene in order to prevent it from being expressed. CRISPR, which stands for Clustered Regularly Interspaced Palindromic Repeats, or RNAi, which is RNA interference. Let's look at each process and decide which is better for our raccoon. CRISPR uses gRNA, or guide RNA, that matches a DNA sequence. The gRNA binds to a protein called Cas9, which pairs the gRNA with the correct DNA sequence. Once paired, Cas9 cuts the DNA, removing that specific gene. The DNA will then attempt to repair itself, an error-prone process that could result in a mutation disabling the gene. A pre-designed sequence can also be added to take the place of the gene that was removed. RNAi uses double-stranded RNA to target specific sites in mRNA, preventing translation of certain genes. The double-stranded RNAs used are siRNA or miRNA. Both bind with proteins to form RISC, RNA-induced silencing complex, and base pair with mRNA. miRNA inhibits translation of a sequence, while siRNA degrades part of the mRNA. Now that we understand the processes, let's look at the pros and cons of each. CRISPR permanently alters gene expression by targeting DNA, and the mutations may not have the desired effect. However, it can target multiple genes at once, and specific sequences can be added to the cell in order to ensure the right expression of the gene. RNAi can unintentionally target multiple similar sections of mRNA, resulting in repression of the wrong genes. However, the effects are not permanent since DNA is not affected. In this scenario, I would choose the CRISPR method. It is more accurate than RNAi, and the addition of the desired sequence can reduce the risk of undesired mutations. This trait is not essential to survival, so permanent silencing of the gene does not put the raccoon at risk. Thank you for watching. All references are in the description. Bye!